Hey everybody. So we are looking at section 16.2, the process of genetic engineering. And so with this, we're continuing along this path of looking at biotechnology and obviously genetic engineering. But in contrast to what we did in 16.1, now we're going to start talking about the futuristic applications of all this. So what is it that we've been doing since we've started to kind of understand what DNA is and how it functions? And certainly since, the human genome has been mapped, where are we headed with that? And what are the tools and techniques that we're using to kind of further assess genetic information and then the proteins that are made from that? So as always, get the heading. And then our objectives for this, I have three for today. So first one here, explain how scientists copy the DNA of living organisms. And we're going to talk about that specifically. That's a technique called polymerase chain reaction. We're going to talk about something called recombinant DNA and how we use recombinant DNA to create things like genetically modified organisms. And then we're going to talk about those GMOs in the context of what we call transgenic organisms. Same thing, just a different term used to represent organisms that have been modified purposefully and genetically. Okay. So in continuing our conversation about genetic engineering and the tools and techniques that we use, you remember what we said before, genetic engineering is going to involve changing and altering a living thing in some way. And in this case, when we talk about genetic engineering, we're looking at actually physically manipulating DNA with a purpose of either investigating and researching something or trying to modify an organism in a way that either allows us to investigate something or in a way that maybe increases things like crop yields, okay? So when we go into actually genetically engineering something, we definitely need to be able to access DNA and alter that DNA in a way. And so when we do that, there are some tools that we talked about in 15.3 that are going to be coming back up here. So restriction enzymes were one of those. If you remember, restriction enzymes, their job was to cut DNA at specific sequences that we call recognition sequences. And those restriction enzymes basically, remember, function as like little tiny molecular scissors. Like they go along and they chop up DNA at those DNA sequences that are specific to that enzyme. And so we can use restriction enzymes, if you remember, to actually excise or remove a gene of interest or just chop up DNA to assess, what the function of that might be. Gel electrophoresis was also covered in section 15.3. And gel electrophoresis is where we're going to separate DNA, remember, according to size. So after we've cut up a DNA sample using restriction enzymes, we can take that DNA sample, put it into a gel, run that electrical current through it. Again, the shortest fragments are going to move the farthest, and the longest pieces stay up at the top. And then based on those size differences, we can identify which genes it was, which segments and fragments of DNA we're actually interested in further kind of pursuing. PCR is where we're going to start here in this section. And DNA sequencing we talked about a little bit also in section 15.3. I have a picture of jellyfish here, two different types, or two of the same type of jellyfish. And what you're seeing is this kind of luminescence. So this isn't fluorescence, it's called luminescence. And I'm showing this here because this is really a protein, this luciferase protein that we call it, this enzyme that causes this glowing kind of phenotype, is something that scientists have been using now to pair up with genes of interest. And what we can do is actually assess when and how those genes are being expressed based on when the organism or the tissue or the cell kind of like lights up like this. And so the expression of a protein would be tied to basically the lighting up of something in this case, just like we're seeing here with these jellyfish. So we've used that protein and also the protein from fireflies and the genes that code for those to kind of watch this process happen in real time. So PCR is right here. And with all of these, by the way, there will be some videos that you're going to need to watch that help to kind of show you this and reiterate this again. Because I don't know that my explaining it always does justice to some of these things, okay? But PCR is what we affectionately call this instead of trying to say polymerase chain reaction. But with that word polymerase, you should recognize that term itself because that was something that we saw in both transcription with RNA polymerase and replication with DNA polymerase. This is essentially that process, but kind of magnified, right? This is kind of like replication or transcription on steroids and exponentially kind of happening faster and more often. So with PCR, we use these again in labs. This is all happening in a test tube on a lab bench like the one I'm sitting at, and in a machine. It's really like a little box-like machine about the size of a tissue box usually. And in that little box-like machine, you can program it. It's essentially a computer that has some different temperature conditions and other types of processes that can happen to kind of mimic and simulate this process as it would happen in cells, but to do it at a faster speed. So what we do in PCR is we take our DNA segment. So again, let's assume what we had assumed back in 15.3, which is that I have a gene of interest. I've cut it out with restriction enzymes. I've run it through my gel. I've cut it out of that gel, and I've assessed it with DNA sequencing to make sure it's the right piece. Okay, if I've done all of that, I really want to make sure that my DNA fragment is not going anywhere, because that's a lot of work, right? I don't want to have to redo all of that every time I want to run an experiment with this. So what I can do is I can copy this DNA segment and then store it, okay? And I'm going to copy it using PCR. So we're going to take our DNA segment. It's just going to be a little tiny amount in solution. And we're going to combine it in solution with several things. We're going to put in DNA polymerase. We're going to put in nucleotides here that are specific for DNA polymerase. And then we're going to program this machine to run a number of cycles. And so usually our cycles are going to be on the order of 25 to 35. And it usually takes 3 to 4 hours to run that many cycles in these machines. But what we're essentially going to do is basically a pattern of heating and cooling and heating and cooling and heating and cooling. So when we heat up DNA, like all molecules, it will actually kind of fall apart, right? We don't want to heat up things too much. But in this case, the double helix will pull apart. And then we have a polymerase here that works exceptionally well at very high temperatures. So that polymerase will come in, and at those high temperatures, we'll actually start to add nucleotides and create copies of what was once together on my DNA double helix to create 2 new strands. Okay, and it adds in these new nucleotides that we've also included in our solution and added to our machine. So we heat, we cool, we heat, we cool, and so on. And so each cycle involves several, steps of heating and cooling. With cycle 2, you can see we go from having, 1 single DNA to 2, and now in cycle 2, we go from having 2 to 4, 4 to 8, we get 16, 32, and so on and so forth, right? So it's like this exponential kind of increase in the amount of DNA. And so again, all that we're doing in this process is we're trying to copy our fragment of DNA so that we have more to work with and we don't just lose all the stuff that we spent so much time trying to isolate, okay? But with those copies, we can use it for cloning, which we'll talk about in just a few minutes. We can use it for recombinant DNA, which we'll also talk about, or DNA fingerprinting and identification, which we mentioned back in section 15.3, okay? This is a picture of a DNA sequencing chart, and this is what I was kind of talking about back in section 15.3 when we were first looking at this process. So DNA sequencing, again, what that's doing is that's telling us the order of nucleotides that make up a piece of DNA. And so, you could do this process at any time once you have enough of a DNA sample to run this. And again, this is also in another machine. This is not something that someone's doing, by hand, but it is something that, you're programming that machine and you are you're including all of the reactants and molecules that are necessary to run it. But essentially, using fluorescent tags and using a polymerase, it's giving us a sequence of letters. And if you can see here, I know they're really small, but all these letters T, A, C, G, and so on, they're all color-coded. So each letter has a specific fluorescent tag, and when that lights up in the machine, it creates these little peaks that we see here. Okay, so sequencing is another technique, it's another tool that we can use to make sure that what we're working with is actually the right piece of DNA, right? It would be problematic if you were working for a long time with a fragment of DNA and you weren't sure of what that fragment actually was, right? So this is kind of used as a self-check to make sure you've got it the right way. Okay, and so this can follow PCR, this can follow electrophoresis alone. It just depends on the quantity, the amount of the sample that you have available. Okay, so like I mentioned with PCR, right? I said one of the applications of having an excess amount of your gene, right? You've copied it and copied it and copied it and copied it, and you have a whole bunch of it now instead of just a little, is in creating something that we call recombinant DNA. So this is kind of that application where we're talking about taking a gene of interest and we're going to combine it with something else in order to put it into an organism. Okay, because maybe we want to see what the protein is that gets expressed, and we want to try and identify what the function of that protein might be. And so if we want to know that information, we kind of have to express it in a living thing. Oftentimes that means we're going to put it into a plant, or we're going to put it into bacteria, or maybe even mice. This is not like we're not going to put it into a human at this point. But we're talking about using these like model organisms to express these traits. So in order to make recombinant DNA, which basically stands for recombined DNA, so we're taking DNA and combining it from different sources, we're going to be combining two fragments or more than two that are coming from different places. So this is a permanent change to the genes that make up an organism when this is done properly. And so you can see here's my restriction enzyme cutting again. So if I have, like here on this side, this is a chromosome from one organism. They're coloring it red to emphasize that. This we would say is a chromosome from another organism. And if we want to join these two together, if you cut them with the exact same restriction enzyme, it'll create what we call sticky ends, which is basically these little overhangs. And when our polymerases and our ligases encounter stuff like this, they're really quick to repair it because they think DNA has been broken. And when they repair it, they're going to very kind of easily and quickly join these sticky ends together. And they're going to join them in the right orientation. Like it can only fit perfectly like a little puzzle piece here. You can't flip it upside down and join it, right? So these sticky ends are super helpful, and they're going to allow us to create this recombinant DNA, which is this down here, that has the gene coming from the blue organism attached to the gene coming from the red, okay? And then we can take this recombinant DNA, we put it in whatever we need it in, right? It can just be in a cell culture, growing in a petri dish, right, in a lab. It can be in a living thing, like we mentioned before, like bacteria or mice or something like that. But regardless of kind of where it's headed, that process of producing it is going to be the same. So one of the things that we like to do in creating these recombinant DNA molecules is to actually take our gene of interest and put it into something called a plasmid. So this is a plasmid. It's a teeny tiny circular piece of DNA. These things are normally found in bacteria, and in bacteria, they kind of add to the genome of those species. What they do in bacteria is they help to provide resistance to things like antibiotics. So we know that we can create bacteria, and this is not a good thing, but we can create bacteria by not using antibiotic medicines properly, okay? And so if we don't use them properly, they can develop resistance. And when they do that, we know that they use these little plasmid molecules to kind of transfer that resistance within a colony of bacteria. So what scientists have done is they've isolated these plasmids, and they've taken these plasmids with specific genetic markers that allow us to ensure that we're adding the right thing in the right position. So what I mean by that is AMP-R is the resistance to ampicillin, which is a type of antibiotic. TET-R is the resistance to something called tetracycline, which is another antibiotic. And so what we try to do in this case, this is the whole plasmid itself, but let's say here is where I want to actually put in my gene of interest. Okay, so I can take a plasmid and cut it with the same restriction enzyme that I used. Let me go back here. Here, this is also an EcoR1 restriction enzyme. And if I cut that plasmid with the exact same restriction enzyme, that's going to create, again, those sticky ends. I can take my gene of interest then and put it basically in close proximity to this plasmid, and the plasmid will kind of take up and join with that DNA fragment. And then we know that our plasmid will have that gene of interest because we can grow these things in a culture dish, like in a lab, in a little tiny plastic petri dish with these antibiotics. So what that essentially means is that if it grows, it's going to have the gene of interest, and it's going to have my plasmid that contains that gene of interest. And so that ensures that anytime I take something like this and then put it into bacteria, I'm going to have my gene in there, and it's going to function the way that it would as if it was in a chromosome from its original place, right? So plasmids are used, again, as another way to create recombinant DNA and ensure that that recombinant DNA can be used and kind of put into bacterial cells. So this is specific to bacteria, okay? When we create organisms, like actual living things, using recombinant DNA, this is one of those processes where we can do that. So this is how we create transgenic bacteria, and we just kind of talked about this whole process. We just didn't see it in this type of image. So one of the things that human geneticists have done is they've been able to take the gene for human growth hormone, and there are lots of people that have issues with maybe a lack of production of human growth hormone. And so there are supplements that we can take now for this. And injections and all sorts of different types of therapeutics. And those are really coming from transgenic bacteria. So we've been able to isolate the gene for that human growth hormone. We've inserted it and created basically this gene, this piece of genetic information, right? By cutting it with restriction enzymes, by isolating it with gel electrophoresis, by copying it with PCR, by confirming its sequence with DNA sequencing. And then this fragment, when combined with a plasmid that's been cut with the same restriction enzyme, EcoR1, that has the same type of sticky ends, can be inserted right here. Okay, and then this can be kind of used to transform bacteria and essentially infect bacteria, but bacteria think that this is normal again because bacteria have these normal plasmids. And when we grow the bacteria in the presence of ampicillin and tetracycline, these two resistance genes that are present in this plasmid, only the bacteria that actually have taken up that plasmid will end up surviving. And so we can pick off in a petri dish little tiny bacterial colonies using little tiny pipettes, and then take those and kind of grow those up and continue this process until we get a large colony of bacteria, which actually doesn't take that long because bacteria grow pretty quickly, okay? Now, what this allows us to do, you may be thinking, well, why in the heck do I want a human growth hormone in bacteria? Well, you don't. There's no real purpose for it. Bacteria don't do anything with it, except for we can isolate that growth hormone that's going to be produced by the bacteria and put it into these drugs that we need, again, for medical therapeutics to treat people that have hormone deficiencies. And so really what that means is that kind of lowers the cost of these types of treatments. We can now create human growth hormone essentially in a lab setting with the help of bacteria, without having to, try and remove that type of hormone product from an actual living, breathing human, right, to give to someone else. So it drives up the production of this. It makes treatment of this disorder a whole lot simpler and easier, and also a lot more cost effective. Other types of recombinant DNA can be inserted into things like plants, and we can make what we call genetically modified plants or transgenic plants. Now, I should have said here, transgenic bacteria is the same as saying genetically modified bacteria. Okay, so you can think about transgenic as meaning genes across, right? Trans is to like move across. So we're inserting genes across from one species humans into bacteria. And so transgenic plants, here we're going to take genes from one organism and move them into something else. So we can create transgenic plants using a type of bacteria that can that usually can cause tumor growth in plants. Okay, I'm guessing you probably didn't know that plants can develop tumors and cancers. Let me go back. And this type of bacteria can actually cause that type of tumor growth. It's called Agrobacterium tumefaciens, okay, kind of like tumor. And that bacteria you can see has a plasmid. What we can do, again, just like we saw on the last slide, is create a recombinant plasmid by inserting our gene of interest, grow that in the agrobacterium cells, and then take that and transfer those to little tiny plant cell colonies. Now, essentially what this is like little stem cells coming from the plants. It can even be little leaf cuttings that we put on a culture dish. You take the bacteria and basically infect those little tiny plant cuttings with that. And then you can create from that entire plants and seeds and so on that will definitely be expressing your gene of interest. And so transgenic plants can be used for different purposes for consumption, like to create increased crop yields in GMO corn and GMO soybeans and GMO papaya that may be like pest resistant or frost resistant or drought resistant, things that plants can't normally survive. This can allow them to survive those if we put the right genes in at the right positions. And this is how you get that group of plants. Now, from this point on, you could essentially just take the plants and continue to inbreed these plants so that they continue to kind of pass down that information. And that's exactly what we see with GMO crops. Okay. Transgenic animals are not simpler. I don't have a ton of information here on this slide for you to write down about that. But all that we essentially do is, again, with that recombinant DNA, we essentially take that DNA and we can inject it into the nucleus of egg cells, which for animals tend to be fairly large, at least under a microscope. They're the largest types of cells we can see. And if we inject them at the right stage of egg development and then fertilize that egg into forming an embryo, that embryo can divide, create a zygote, and then a whole living thing that expresses that DNA of interest. Okay, so we'll do this with mice, with fish, with simpler organisms like that, but we don't typically see this happening in humans, that's for sure. All right, another genetic engineering method is called CRISPR technology, and this is the most recent addition to our list of new methods of biotech procedures. So CRISPR is a really long acronym for Cas9 assisted. It goes on and on and on, and I cannot tell you exactly what it stands for because it's complicated and it's new, but just know it's called CRISPR. Okay, you do not need to know what that acronym actually stands for. You can look it up if you would like to. But CRISPR technology is super cool because it actually allows scientists to change base sequences, including in humans, without taking DNA from another source, without having to create actually recombinant DNA, without having to go through all of these extra steps that we see in the process of making transgenic bacteria and transgenic plants and transgenic animals. In this way, what this essentially means is that scientists have a means by which we can actually directly edit a gene in a living thing at that specific time. It doesn't have to be early in development. It doesn't have to be at a certain stage. It could be like literally right now, something that could be edited in my body using this technology. And so what this requires is an enzyme called Cas9. And Cas9, you can see they have little tiny scissors here and what they're labeling as our active sites. So Cas9 does a really great job of chopping up DNA. And kind of similar to restriction enzymes, it's going to chop up DNA at specific sites. Now, the site that's identified for this is based on a target RNA sequence. So this target RNA sequence is something that for a scientist, they would identify, they would create in a lab, and then they would combine with Cas9 along with their DNA of interest. And that would allow Cas9 to come in and essentially bind to and start interacting with that target DNA here. It's going to cut that DNA based on this target RNA sequence, so at a specific position. And then it's going to insert in that position a complementary strand using random bases. Okay, so if it creates this double-stranded cut like we're seeing right here, it can just put random bases in that position. And this is essentially what we call a gene knockout. You can take a gene of interest, have the nucleus randomly repair that gene, and that's essentially probably going to turn it off. And then from that, you could assess, okay, what was the impact of that? What were the effects of that? What must that gene have been responsible for doing? Okay. Now, in addition to that, what makes this even more helpful is that we actually can use this in a therapeutic sense. So we can actually take our cut DNA, our original genomic DNA, and insert to that a new DNA sequence right here. And that new DNA sequence could contain, for example, let's say I have a mutated gene here that causes human growth hormone deficiency, right? If I cut that DNA at that position and essentially, break apart that gene, but now come in and actually insert the correct copy of that leads to not the mutated, form of that gene, but the normal one that produces normal amounts of human growth hormone, I have the ability then to essentially edit that gene, correct that error, and for the individual, that can lead to better outcomes, right? It could totally rid the body of a specific disorder. Now, the catch would be you'd have to be able to do that in all of the cells of the body, right? Which is actually not completely ridiculous to think of. It is something that is possible. So Cas9 has been used recently to start going through these gene therapy procedures to try and correct disorders specifically in humans. It's a pretty cool technique without a lot of controversy, I should add. Okay, and I've got just, I think, 2 more here. So when we talk about doing all of this, right, one of the applications is also in cloning. And cloning is something I'm sure you've heard of before. With a clone, you can clone basically anything in a lab, from bacteria to plants to mice to humans and so on. But a clone is essentially an identical copy of a gene or an organism. And yes, when we copy a gene using PCR in that little machine that I said sits on the lab bench and goes through all these cycles of heating and cooling, that's called cloning. So it's not simply, this super controversial stuff where people, think about these sci-fi type effects. Cloning can be as simple as just copying a gene. Okay, so we routinely create these in biology labs to research disorders, to improve quality of life. If you're going to talk about cloning an entire organism, that's a whole lot more complicated than just cloning a sequence of letters, right? And there's a lot more involved there. But for the most part, cloning is something that is kind of frowned upon if you're talking about cloning an entire organism. But cloning of genes is something that's super acceptable and is done pretty routinely in every biology lab probably in the world. So this is that process, and you can see there's a lot of similarities here to what we've already been talking about. So here's my bacteria, and I am going to use bacteria as part of this. I've got my plasmid. Gene of interest here is this little tiny black dot. So again, you cut it out, run it through a gel, you copy it, you sequence it, make sure it's all well and good, and then you insert that into a plasmid using the same restriction enzyme cuts. You grow that plasmid in bacteria, you isolate that bacteria, and then you can do all sorts of different things, right? We can infect plants. We can research a gene by putting it into other types of bacteria and looking at the impacts of that. We can isolate the protein that's being made, like we saw human growth hormone, and then we can, do other medical therapies with some of this stuff. So really the options are kind of limitless when we talk about cloning of a gene. Okay. Now, cloning of an animal, I said, is one of those things that gets a little bit more risky and complicated. So when we think about cloning of an animal, one of the first animals to be cloned and cloned successfully was a sheep, and that sheep was named Dolly. And this was years and years, probably it has to be decades ago now. This was over, I think, in Ireland or something, over out in Europe. But Dolly the sheep was cloned kind of using this process. So what they do is they take a female sheep and they obtain an egg. So they have an egg donor is what we call her. They would remove the nucleus of that egg. And then we have a cell donor where we're going to remove the nucleus from that cell. Okay. Now the cell donor is what we're actually trying to copy, right? So we're going to take the nucleus out of who will eventually be kind of the foster mom, and put into that cell the DNA, the genetic information from our donor organism, from the one that's actually going to get cloned. So the DNA goes into the mom cell. That cell is fused, right, with this nuclear information, right? It's fertilized, and then the embryo grows until we eventually can implant that into the mom again here, the same one that was our egg donor. And then the mom gives birth to essentially this same lamb, okay, the one that we originally started with, the same sheep. So this is kind of the process of that. Now, the catch is, again, you have to have the ability to gestate this. You have to have a female that can carry that offspring, right, to term and then have a live birth. But this is a pretty cool technique that, again, is strictly lab-based. We're using tools that have to be employing lab technologies in order to get this done, okay? But essentially, you're just taking genetic information, putting it into the egg from our donor, and then allowing that to grow into a brand new organism. So something that's definitely being done. Um, the applications of this one are a little bit more, again, controversial because you're talking about creating a copy of a life form that was already existing and maybe should not be existing anymore. But it's there. So it's definitely interesting, at least, to talk about. Okay. So that's all I've got for 16.2. I know this was a long one. So let me know if you need anything, if you have any questions. Otherwise, I will see you for 16.3. Bye, y'all.